So strategies to manipulate the microbiome, you know, they're really still emerging, as you just saw in this presentation, this great presentation by Denise. And we're producing promising areas of research. So on behalf of Howard and Van and myself, we really do want to thank the Biopharma Research Council and the National Cancer Institute for the opportunity to share an overview highlighting some of the areas that look like they have exciting directions and challenges in altering the microbiome being addressed, especially my talk's going to really focus on probiotic, prebiotic research within and beyond the NIH, and really inputting this type of overview together. It really is an opportunity that we don't get too often to just sort of look ahead and consider the possibilities of next generation technologies during the in terms of driving the science now with probiotic, prebiotic foods and pharma approaches. So probiotics, first of all, can be considered both exogenous and indigenous to the biological system. And these are live microbes that when they are being used in manipulation therapies, they're considered to have a beneficial effect if given in appropriate amounts. They also ferment carbohydrates and have little proteolytic activity. This was a definition that's been expanded upon from the World Health Organization and the Food and Agriculture Organization of the United Nations. So mechanisms of probiotic action really in the context of the microbiome are being examined in all of the organ systems, but in the gastrointestinal tract using both in vitro and animal models. And these do include remodeling of microbial communities and suppression of pathogens, suppression of pro-inflammatory factors, effects on the epithelial cell differentiation, and proliferation and promotion of intestinal barrier function. So the most extensively studied probiotics, as most of you know, are the lactic acid bacteria, your lactobacillus and bifidobacterium, and some yeast as well. Prebiotics are considered indigestible substrates that reach the colon. They're available for fermentation by both commensal and pathogenic bacteria. So substrates that are widely studied as prebiotics are fructans, inulins, fructo and galacto-oligosaccharides, and resistant starches. Now, this is a schema that was first developed by, as I say, the World Health Organization and the Food and Drug Administration, along with other regulatory bodies and the Food and Agricultural Organization Committee. And it really was established to look at standards of strain specificity and dosing accuracy, which we're really now able to do because of the high throughput technologies that are available and weren't available previously. And this particularly has helped us to look at genotypic and phenotypic identification and taxonomy that was really not possible at the strain level previously. It's also provided us a means to really look at the potential for antibiotic resistance traits. So although clinical safety and efficacy studies are well on the way, most of the current research continues to look at the metabolic and microbiomic functional properties being examined in vitro artificial gut systems, in germ-free and autobiotic animal models, and as well as in predictive in silico models. And really, in essence, you need all of these from your in vitro models to in vivo and animal studies to really look at the functionality and benefit and substantiate that in terms of manipulating the microbiome. And I think some of the promising areas are certainly in terms of metabolite production of these microbes. It's really an incredible treasure trove of new drug. The formation of biofilm capacity or not, the competitive evolution of and competitive exclusion in pathogenicity and co-aggregation dynamics, and the production of antimicrobials. So one of the even more promising technologies is metabolomic footprinting. And this gives us the capacity to, in multi-scale networking, to really begin to look at both the genetic and metabolic changes in response to perturbations, in response to manipulations like antibiotics, vaccine, as well as nutrients and other drugs. Three major developments, however, have really promoted the lactic acid bacteria as a potential probiotic that can manipulate the microbiome for beneficial effect. The first is the genomic revolution. When the genomic revolution came upon us, lactic acid bacteria had already been being really well studied and characterized, a lot of the reason being the food industry, that it had already developed very large culture banks. But with the genomic revolution, we now have 25 genomes that are completed in nearly another 100 that are nearly complete and currently available for next generation. All of these include one aspect or another of next generation technology screening. A second reason why I think this has really expanded as a field is because of the next generation technology sequencing. This has really allowed us, again, an enormous high throughput capacity to really look at genomics and metabolomics and other interaction effects between various body sites and the gut. And a third reason is because that we are really moving very rapidly into systems biology and synthetic biology approaches to integrating all aspects of metabolism, genetics, and functional properties. And certainly approaches with probiotic on the microbiome are one of the most promising areas. So modulation of gut microbial metabolic functions using pro and prebiotic and symbiotic approaches have these types of extensive multi-compartmental modeling approaches, as you see here. And it really, again, it allows us to take body fluid and tissue and to look at it in multi-dimensional ways and look at microbial changes that potentially impact human body fluids and host lipids, carbohydrate metabolism, and every mammalian biological system that's been studied to date. It's very exciting. Now, in terms of this point of this whole overview, what synthetic biological approaches could be applied to probiotics, particularly these lactic acid bacteria strains, has been a major interest among researchers as well as drug manufacturers. And one avenue is to build upon some of the functions that are present and can be optimized given the profound knowledge that we actually have about lactic acid bacteria in these culture libraries. In terms of producing vitamins, vitamin B12, vitamin K, short-chain fatty acid production that you see in Lactobacillus rhamnosus GG and L. reuteri, L. acidophilus, and many of your bifidobacterium strains. And so advancing the understanding of such metabolic network functions will really require continued investment on the part of both public and private. And one of these avenues that has been used is the Bridging Interventional Development Gaps program. And this is a program at NIH where it enables NIH researchers to provide preclinical services that really include test systems and pharmacokinetics and toxicology studies for potential therapeutic applications. And although it's still in its infancy, the effort to encode metabolic footprints such as sucrose-nisin transposon and derivatives have been recently discovered. And a lot of you know the nisin-induced control expression systems are now actually used worldwide for controlled gene expression for regulatory circuits. So fundamentally, strain improvements of lactic acid bacteria are also very important in moving along in this continuous fashion. And some of the strategies, again, following the World Health Organization and FAO committee, is to resequence technologies that can detect multiple mutations, and by this natural conjugation, mutational evolution, it avoids the genomic modified organism approach and is examining metabolic pathways relating to their functional interdependence, the robustness of selected strains, and enhanced health benefits. So a very exciting avenue that's working in various branches. So to just move a little to the biological system side, humans stand at the end of a mammalian long evolution with differences in human milk and mammalian milk systems. And they're distinct in terms of their volume, the number of human and bovine, for example, milk structures, and the complexity of milk oligosaccharides present in these milks. While microbiome manipulation may have benefits at any age, if we were really to say where has it its most potential, it most likely will be in the coming years in terms of the critical window of developmental biology. And so this really allows the potential for a self-completion of postnatal programming that goes on. Glycans are really a complex chain of carbohydrates, also referred to in oligosaccharides, that are abundant in human and mammalian milks. And one of the interesting evolutionary concepts is that the fucosylation really increases as you move from the mammalian species into human, and sialylation decreases. And exactly why and how this happens, we don't know, but one of the really interesting questions is that whether it's a human mother or another mammalian maternal dam, the human milk oligosaccharide or the milk oligosaccharide glycan doesn't really seem to offer any nutritive value to the offspring without being digested first by the microbe. Human being cannot digest the oligosaccharide in breast milk. It must be digested, degraded by the microbes in the system. And what we see for many decades now, but even more so with the high throughput, is that bifidobacterium and lactobacillus are among the microbiota that become very prodigious in human systems. And there's one particular bifidobacterium strain the Bifidobacterium longum subspecies B. infantis, as we call it now. But B. infantis has this very unique capacity to digest any oligosaccharide, any of the more than 400 in human milk. And so it's really looked at now as a possible avenue for new infant formula. And this unique consumption of human milk oligosaccharides by Bifidobacterium infantis allows it to really benefit the neonate, and further studies have shown that it actually improves intestinal permeability, excessive inflammation, and immune response protection. So it's somewhat of a compelling case of coevolution of a lactic acid bacteria strain, a probiotic strain, if you will, and the human host. And recent comparative studies of these various human and bovine and other mammalian milk oligosaccharides has shown that these impart this type of protection in both the normal and preterm model systems. So it has some application, and we think a lot of promise for areas of research with neonatal premature infants who are at great risk of necrotizing enterocolitis. Now, another open question that's being examined is how can probiotic bacteria convert their health benefits to the host? And again, recent evidence suggests that bacterial cell surface molecules and macromolecules are key factors in a probiotic microorganism and host crosstalk. And this signaling that's going on is an area of intense investigation, as they can interact with the host pattern recognition responses of the gastrointestinal mucosa. One area of investigation at NIH and in the broader research community is really being highlighted in documenting signaling interactions of surface macromolecules of probiotic bacteria, including some of the pili of Lactobacillus rhamnosus JG, and also intercellular adhesion molecules. So these distinctions and parallels are very interestingly being studied between the presence in an array of commensal organisms as well as pathogens to see if the same types of host microorganism crosstalk, where are the parallels and where are the unique distinctions. So extensions of this research and other laboratories suggest that there is truly a single layer of epithelial cell lines that the small in the large intestine and the function of the barrier function benefit from, where these commensal bacteria kind of layer it from the rest of the body and peritoneum. And that toll-like receptors on intestinal epithelial cells by bacterial products promote epithelial cell proliferation, secretion of IgA into the gut, and expression of antimicrobial peptides. So this established a microorganism-induced program of epithelial cell homeostasis and repair in the intestine. And we really see here that this regulation of this process can actually result in chronic inflammatory and overproliferation of the repair process. And this dysregulated toll-like receptor signaling by intestinal epithelial cells may explain how colonic bacteria and inflammation promote colorectal cancer that requires much more intensive investigation. I don't have time to go into these in more depth than that this morning. So, but in extending this just a little further, gut bacteria host signaling is continuous, it's reciprocal in the pro-cancer macroenvironment, we see beneficial microbes trigger IL-10-mediated GI tract neuronal networks and lower systemic inflammatory tone and upregulate hyperthalamic targets, including perhaps oxytocin, constituting a gut systemic stimulation of CD4 lymphocytes, including regulatory T cells that suppress and promote and have either they promote it or they protect from it, or there's no effect, depending on the timing and a priori exposure to gut bacterial antigens and presence of other factors. So this places neoplastic development and growth into a broader paradigm in the microbiome, where we are beginning to look at it as what they call a holobiome, and it's really consisting of the mammalian host and its resident microbes, and that cancer as a macro environment might actually be considered such that the microbes can be engineered for sustained improved benefit. Taken together, the perspective really presents the possibility that next generation type technologies do include probiotic strategies for fighting cancer and for promoting overall good health and longevity. Now, probiotics also increase, as I mentioned, antimicrobial secretion systems, and one is defensin from intestinal epithelial cells and directly block signaling pathways from pathogens. Significant developments here have emerged since the first description of lactococci, and these lactococci really were used as an oral vaccine approach with specific helper functions. Orally, as Denise mentioned, we see orally delivered vaccines having been historically shown to underperform in developing countries. And some of this is, of course, the heavy burden of nutritional deficits and diarrheal disease in developing countries. So one of the challenges is how can we optimize gut barrier function and immune responses in these phenotypic populations and locations geographically. So supplementation with specific strains of probiotics have been shown to have modulatory effects on intestinal and systemic immune responses in animal models of allergic disease. And just very recently, we see an anti-helminthic drug being used in cholera vaccine, where that anti-helminthic was given to the population 2 weeks before the cholera vaccine was administered, and there was a reduction of cholera type B toxin in these subjects with respect to specific IgA levels as well. Mm-hmm. Oh, 2 minutes? Okay. So resilience is another concept that we're just beginning to really look at. What does it mean to be a resilient host? And it can be perturbed almost with anything: diet, antimicrobials, antibiotics. But resilience is the capacity of the system to absorb disturbance and reorganize while undergoing change, so as to really retain essentially the same function, the structure, the identity, and the feedback. And I will just sort of skip over that and use it in another slide. One of the other areas with probiotic is you either have the single system or the biofilm. Biofilm by pathogens and biofilm capacity following, in fact, vaccine administration and infectious disease is one of the arsenals for taking on sort of a community behavior so that the pathogen can persist and it very much depends on the timing and the aging. Probiotic may be a strain intervention strategy that we can use in NICUs and in other environments where biofilm pathogenicity is highly prevalent, and we can help to dissolve the ability of the biofilm pathogenicity to persist. And I think this last slide, I just want to give you a little bit of thought on the emerging and exciting concept of the microbiota gut-brain axis that suggests that the modulation of gut microbiota by probiotic and prebiotic strains and strategies is another avenue that is being explored for novel therapeutics. And one of the world's best known, and I think many of you know this, is John Crean, and he will be coming to NIH on October 6th, and we certainly hope you'll enjoy the lecture. But John's world has suggested that you cannot be a human being and be socialized, that the microbes really depend on us it's part of the socializing mechanism, and the germ-free mice that are not propagated with animals, you'll notice in germ-free mice they have very antisocial, they have high anxiety behavior models, and John's manipulated a lot of this. I don't want to try to go into that in any great detail, but we think that future research is likely to give very special attention to the gut-brain axis and how the metabolomics plays into the signaling neurogenically, immunologically, and metabolically. And so in the next level of systems design, you may see quite a bit of attention in this area. Thank you very much.
So I'll start off. And so, Linda, if you go to Paris and you have yogurt, you're supposedly getting L. acidophilus and other bugs in the yogurt. If you go to Seattle and get yogurt, you're supposedly getting the same bugs. But do we really know they're the same bugs? And what do you think the industry has to do to standardize and ensure that what people think they're taking is what they're really taking?
Yeah, thanks. Thanks, Howard. So whether you're in nutritional or in drug development, I think the bottom line is that we really need to standardize these collections with genotype and phenotypic taxonomy and identification so that we know what's there is there at the strain level, and that these relational databases need to really be quite expansive in terms of, again, the systems approaches with metabolomics and various functional properties.
Okay, other questions? So is there, okay.
You mentioned genetically modified organisms and coming up with sort of non-GMO ways to make mutations, which really makes you a GMO. So one of the things I think that perhaps NIH and FDA could maybe even take a lead on is this sort of dichotomy between the U.S. and Europe about what it really means to be genetically modified. We can make mutations in our probiotic that you could never distinguish from a natural mutation, yet the U.S. is okay with that, but Europe says you used a plasmid, even though the plasmid is completely gone, so you're GMO. So I think that that's something going forward that there needs to be a lot of discussion about. Anyway, it's just a comment.
Yeah, I think it's an excellent comment, and it's interesting. I just flew in at midnight last night, which is part of my confusion today with Millingate, and I was just at a conference where this was a big topic of conversation, and the Europeans and the Americans were very much discussing different aspects of lots of areas, not just that, but I think it's a very important topic in this field.
So another question I have is, so there have been a number of papers from countries such as Korea that report novel probiotics with novel properties, but it proves impossible to get those organisms to study. What do you think the research community and regulatory communities have to do as a whole to make these organisms that supposedly are reported to have useful properties available to the broader community?
Yeah. I think that we're hitting a point where the microbiome, mostly everyone in the room would agree, this is an extremely expensive area of research. So in a way, I want to say probiotics and prebiotics benefit from it because they hitchhike in terms of really being able to look at microbiome in the context of a target organism within the milieu of the microbiome communities. But I think that the work, just as the Human Microbiome Project really began as an international consortium effort with MetaHit in Europe, working with the U.S. and particularly the NIH Human Microbiome Project. Again, from yesterday's meetings, I can certainly see it's certainly moving in that direction, that we're going to see far more international cooperation, international interface of databases and standards than we probably have been able to touch upon in our own research experience from our own individual libraries. But it's definitely happening, Howard.
Okay. Well, thank you very much. So hopefully our next speaker is here.